Hello and very good evening to the students who have joined. Can you just confirm in the chat box if you can hear my voice properly and you can see the video and the PPT as well? Okay, so we will wait for 5 to 6 minutes and then we will start today's session. Okay, so very good evening. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is the week 10 live session for the NPTEL course of Medicinal Chemistry. In today's session we will discuss about the week 11 course contents. Okay, the course contents which are included in week 11 and that related questions 10 to I think 20 questions MCQs we will discuss today. And the questions will help you to understand the course better. Okay, so the questions will help you to understand the course better, and it will also help you to know what are the things which you have to put your strength in. And one more thing I want to tell you that there may be some internet disruption today as well. So if there is some internet disruption, I want all of you to wait for some times, don't leave the class, okay, and wait for some times. I will definitely rejoin back. And if you have any problem for hearing my voice or looking at the PPT, you can just write in the chat box, okay. After each and every question I will wait for some time, and I want you guys to answer that question in the chat box. It may be wrong, it may be right, but answering the question is very important. Okay. So, after every question please try to answer the question, no matter if it is right or wrong, but at least you, all of you should try. Okay. So, I think we can start today's session, and in between okay, I think I will wait for 2 more minutes. Okay. And in between then we will start today's session, and eventually when other students will join I will just add them up. So, I think it will be better if I wait for 2 more minutes. So, I think my voice is audible and you can see the PPT, right? If you have any problem, just do let me know in the chat box. And as I said, if there is some internet problem from my side, so I want all of you to just wait for some time. Okay, I will surely rejoin back in 2 to 3 minutes, but don't leave the class in between. So, you can see the PPT, right, and you can you can hear my voice clearly. Okay, okay, very good. So, I think we can just start our class then. Eventually when others will join I will just let them okay. So, today it is week 10 live session, and our today's topic is included what you have read in your week 11. Okay. So, your week 11 it has some antiviral drugs, some anti-cancer drugs, and related to the question will be related to those topics. Okay. So, try to answer the questions as I show you, and if it is right or wrong it does not matter, but try to answer the question in the chat box. Okay. After each question I will wait for some time, and after looking at your answers we will I will show you the correct answer and we will discuss that. Okay. So, the first question for today's session is okay. So, there is a typographical error, I will just correct it later. Okay. So, it will be problem number 1. So, which of the following statements is regarding the virus is false? Okay. Number A, viruses are submicroscopic organisms. Number B, viruses only grow and reproduce inside the host cells. Number C, COVID-19 is a viral disease. And number D, viral infection is confined only to a particular part of the body. So, which of the following statements regarding the virus is false? Number A, viruses are submicroscopic organisms. Number B, viruses only grow and reproduce inside the host cells. Number C, COVID-19 is a viral disease. And number D, viral infection is confined only to a particular part of the body. I want everybody to try and which of the following you think may be false regarding the virus, you can write in the chat box. Okay. So, do we have any correct answer? Number D, number D, number D. So, till now everybody thinks it is number D, right? Okay, let us check out what is the correct answer. Okay, so the correct answer is number D, viral infection is confined only to a particular part of the body. Okay, so first let me just correct this thing, I just have a typographical error. Okay. Okay. So, correct answer was, as I said, number D, viral infection is only confined to a particular part of the body. This statements regarding virus is false. Now, let us look at the viruses and what is a virus. So, it is a pathogen or a disease causing agent not considered living because it cannot reproduce of its own. Okay. So, viruses, as we all know, unlike bacteria, so they cannot reproduce of their own, that is why it is not considered as a living. Okay. Now, what is the difference? What are the differences between the virus and a bacteria? So, viruses are not living organisms, but bacteria are living organism, and the reason is viruses cannot reproduce of its own, but the bacteria can. Okay, then this is the major difference between a virus and a bacteria. Viruses only grow and reproduce inside the host cells they infect. When found outside these living cells, viruses are dormant. Their life therefore requires the hijacking the biochemical activities of a living cell. But bacteria are living organisms that consist of single cell that can generate energy, makes its own food, move and reproduce, typically by binary fission. And this allows bacteria to live in many places like soil, bacteria, plants and the human body, and serve many purposes. So, the main difference which between the bacteria and the viruses is that the viruses only grow and reproduce inside the host cells of the infect. Okay, outside the host cell it does not have have the capability of the reproducing and growing of its own. Okay, but the bacteria, it can actually grow and reproduce of its own. That is why we can find bacteria in any places like soil, plant and human, any part of human body and many things, because they can survive of its own. They does not need a human body or any host to reproduce and make their own food. Okay, that is why the viruses are not considered living, but the bacteria are considered as a living organism. Viruses are submicroscopic. Bacteria is a giant compared to the viruses. So, the volume as you can see, the size or volume of the bacteria is large compared to the viruses. That is why viruses are called the submicroscopic organisms. Okay. A viral infection is systematic. Viruses infect a host cell and then multiply by thousands, leaving the host cell infecting other cells of the body. Bacterial infection is usually confined to a particular part of the body, described as a localized infection. Infection may be caused by the bacteria or by the toxin, like endotoxins, which are produced by the bacteria. So, viral infection, it is not confined to the particular part of the body. It may start at some place and with a particular host cell, but soon it will just start reproducing and start infecting other cells of the body, infecting our body as a whole. But the bacterial infection, it is too much systematic and a localized infection. So, if you have some bacterial infection in a particular part of the body, let us say on your skin. Okay. So, it will confine to that part, okay. It will not go to other part of the body. Systematic diseases caused by viral infection include influenza, measles, polio, AIDS and COVID-19. Bacterial diseases include pneumonia, tuberculosis, tetanus and food poisoning. So, these were some examples for the viral and bacterial diseases. Okay. So, this was all about the viruses. So, I think it is clear to all. And as I always say, if you have any doubt, any query after any question, you just can write in the chat box. Okay, after each and every question I will try to answer that question. Okay, and after each and every question I also want you to answer that question. Does not matter if the answer is correct or wrong, but try to answer this question. So, this question was all about the viruses and the key differences between viruses and the bacteria. Okay. So, now I will move toward the next problem. So, problem number 2, okay, there is some typographical error thing. Okay, I will just correct them later. Okay, so the problem number 2, it says that which of the following enzymes is responsible for processing HIV proteins during the production of the new viruses. Number A, integrase, number B, protease, number C, reverse transcriptase and number D, DNA polymerase. So, which of the following enzymes is responsible for processing HIV proteins during the production of new viruses? As we all know, HIV, which is actually the HIV protein or human immunodeficiency virus, this is responsible for the disease named AIDS, and which is very famous viral disease. Okay. So, there are very complicated mechanism by which this particular virus works, and you have to identify the correct enzyme which is responsible for processing HIV proteins during the production of new viruses. Your options are number A integrase, number B protease, number C reverse transcriptase, and number D DNA polymerase. It is your problem number 2. There are some typographical error in each slide, I think, regarding the problem numbers. I will just correct them before uploading this PPT to the website. Okay. So, do not worry about it. So, write in the chat box which of the following you think is the correct answer. Okay, so is there any answer for this question? Okay, number B and number B. So, we have till now 2 answers, right? Okay. Okay, just give me 1 minute. I will just stop presenting and I will just correct the numbering of the slides. I think there is some problem, and just give me 1 minute. I will just correct them and we will start. Okay. Others also try. Which were the options? I think you know. There was integrase, protease, DNA polymerase, and reverse transcriptase. Okay. Which of you think is the correct answer? Okay, now it is correct. Okay. Yeah, I will just reshare them, don't worry. I was just correcting the numberings. Just give me 1 minute, there is some problem. Okay, you can see the slides now, right? Is the presentation is visible now? You can see the slides, right? Yeah. Okay, so we were on our second question, right? So, the second question, it was which of the following enzymes is responsible for the processing HIV proteins during the production of new viruses, and the options were number A integrase, number B protease, number C reverse transcriptase, and number D DNA polymerase. Okay. So, in the chat box we have 1 or 2 answers for it. So, let us check out what is the correct answer. So, the correct answer is yes, number B protease. Okay. So, let us discuss about the replication cycle for this particular some viruses. Okay. So, what is the about the replication cycle of the virus? So, infection begins when the viral spike or S glycoprotein attached to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor attached spike protein. Depending on the host cell protease available, cleavage and activation allows the virus to enter the host cell by endocytosis or direct fusion of the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated and its genome enters the cell cytoplasm. On the coronavirus RNA genome has a 5' methylated cap and 3' polyadenylated tail, which allows the RNA to attach the host cell's ribosome for translation. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own proteases which cleave the polyprotein into multiple non-structural proteins. A number of non-structural proteins coalesce and to form multi-protein replicase transcriptase complex or RTC. The main replicase transcriptase protein is the RNA dependent RNA polymerase. It is directly involved in the replication and transcription of the RNA from an RNA strand. The replicated positive sense genomic RNA becomes the genome of the progeny viruses. Progeny viruses and then released into the host cell by the exocytosis through the secondary vesicles. Infected carriers are able to shed viruses into the environment. The interaction of coronavirus spike protein with its complementary cell receptor is central in determining this tissue tropism effectively and species range of the released virus. Coronavirus mainly target epithelial cells. They are transmitted from one host to another depending on the coronavirus species by either the aerosol, formite and facial oral route. Human coronavirus infect epithelial cells by respiratory tract, while animal coronavirus generally infect epithelial cells of the digestive tract. SARS coronavirus, for example, infected via the aerosol routes and of the human epithelial cells or lungs by binding to angiotensin receptor converting enzyme to receptor. So, this was about the replication cycle and the example is given is the coronavirus. Okay. But still this is the basic replication cycle which is followed by all viruses including the HIV proteins also. Okay. So, the enzyme which is responsible for processing HIV proteins during production new virus is the protease, and that is why HIV proteases are a very important drug target for developing new anti-AIDS drugs. Okay. So, if the protease, the drugs which are used as called the protease inhibitors. Now, proteases are the enzyme that cleave the protein molecule into small fragment, and HIV protease is vital for both viral replication within the cell and release of mature viral particles from the infected cell. Various protease inhibitors are developed for the HIV treatment. Some of the FDA approved drugs are the saquanavir, indinavir, atazanavir, ritonavir, etc. Okay. So, this was about the protease inhibitor and a brief discussion about the replication cycle of the viruses, how the viruses replicates. Okay. And I think you do not need to memorize all of these things. I just gave you an overview of how the viruses replicate, because so that it is understandable to you how the things work by. Okay. So, this is also only for your knowing. I do not think this is important for how the viruses replicate for your exam purpose also. But the protease inhibitors are a very important topic which is important for your class also, for your course also and for your exams also. Okay. So, I think this was your problem number 2, and okay, now I will just move toward the next problem, and as I say there may be some internet disruptions. So, if you have any problem for listening my voice or not, you are not able to see the PPT, you can just directly write me in the chat box. Okay, and if sometime my voice or the PPT got disconnected, just wait for 2 minutes, I will just figure it out. Okay. So, the problem number 3, it says that what sort of heterocyclic ring is present in the histamine? Number A, imidazole, number B, thiazole, number C, pyrazole and number D, oxazole. So, which of the heterocyclic ring is present in the histamine? I want everyone to try to answer this question in the chat box. It does not matter if the answer is correct or wrong, but try to answer it. Which of the following heterocyclic ring is present in the histamine? Imidazole, thiazole, pyrazole or oxazole? So, is there any answers for it? Number A. So, we have two answers and two of them is number A. Okay, let us check what is the correct answer. So, the correct answer is yes, number A, imidazole. So, let us took a brief overview about the azoles also. So, this is the histamine. Okay, and we all know histamine is very much related to the allergies of our body. It is a compound which is related to that thing, and the ring which is present in the histamine is the imidazole ring. As you can see, this is this 5-member ring, it is the imidazole ring. Okay, and in this 5-member ring the nitrogens are at 1 and 3 position and it is called imidazole. So, azole is a class of 5-member nitrogen heterocyclic ring compounds containing at least one other non-carbon atom of either nitrogen, sulfur or oxygen. A majority of compound produced by nature have heterocyclic ring as a part of their structure. Many heterocyclic rings are found as a key component of the biological system, and these rings are named as you can see here, this is pyrazole, this is imidazole, this is thiazole, this is oxazole and this is isoxazole. So, these rings are very important, as I can say as I can tell you that azoles are a very important class of heterocyclics which are included in the human body also. Okay. So, many are the human body and many of the parts which have the heterocyclic rings, and you will see azoles have the very much prevalence in also in the drugs also. So, many are azole, there are many drugs which have this azoles as a pharmacophore, mainly as I think you have heard of the antifungal agents, okay, antifungal creams like ticonazole, fluconazole, and these kind of drugs also have the azole component. And so, remembering the names and the structure of azoles are very important if you are reading the medicinal chemistry. Okay. So, as you can see, azoles are actually 5-member ring. So, if there are 5-member ring heterocyclic and it contain at least one other non-carbon atom of either nitrogen, sulfur or oxygen. So, as you can see, if the nitrogen there are 2 nitrogen and nitrogen are at 1 and 2 position like this, 1 and 2 position. So, it is called the pyrazole. Okay. And if the nitrogen is present, though 2 nitrogens are present in the 1 and 3 position like this, and this is 2 carbon and 3, then it is called imidazole. If there are one S and N are present in 1 and 3, this is called thiazole. Oxygen and N, this is called oxazole. And if oxygen and N, just like pyrazole, nitrogen at 1 2 position and oxygen and nitrogen is 1 2 position, it is called the isoxazole. So, remembering the names and the structures of azoles are very important, and I think it is not very much complicated structure. If you just read for few times, you will just remember it easily. Okay. So, this is all about the azoles. Now, we will move toward our next problem. So, problem number 4, it says that steroid hormones combined with intracellular receptor to form structures that control the expression of genes. What are these structures called? Number A, nuclear translation factors. Number B, nuclear transcription factors. Number C, nuclear replication factors. And number D, nuclear reproduction factors. Which of the following you think is the correct answer? Steroid hormones combine with intracellular receptors to form structures that control the expression of the genes. What are these structures called? Nuclear translation factors, nuclear transcription factors, nuclear replication factors, or nuclear reproduction factors. Is there any correct answer or any answer for this problem? Number B. So we have 2 number B's. Okay, I want others also to try to answer. Okay. Let us check what is the correct answer. So the correct answer is very good. Number B, nuclear transcription factors. So transcription factors of eukaryotic cells. So activator proteins. Okay, so if somebody wants to join. So transcription factors of eukaryotic cells. Activator proteins bind to the pieces of DNA called enhancers. Their binding causes the DNA to bend and bringing them near a gene promoter even though they may have thousands of base pairs away. So this is the enhancers and this is the activator protein, and as you can see the other transcription factor proteins are these, and that's how they come closer. Other transcription factor proteins join the activator proteins, forming a protein complex which binds to the gene promoter. Okay, and as you can see, this is the gene promoter and this is the RNA polymerase. Now this protein complex makes it easier for RNA polymerase to attach to the promoter and start transcribing a gene. So transcription, which is an important thing here, and protein complex makes it easier for RNA polymerase, this is the RNA polymerase, to attach to the promoter, and this is the promoter as you can see here, and start transcribing a gene. Okay. An insulator can stop the enhancer from binding the promoter if the protein called CTCF, named for the sequence CCC-TC, which occurs all in the insulator, binds to it. So as you can see, there is an insulator presence here which is CTCF and which is CCC-TC binding factor. Methylation, the addition of a methyl group to the carbon nucleoside prevents CTCF from attaching the insulator, turning it off, allowing the enhancers to bind the promoter. Okay, and this is the insulator, how it is works. So that's how the transcription happens, and transcription factors are the main, I think, intracellular receptors which are formed after the steroid hormone combined with the intracellular receptor of the structure. Okay. So this was all about the nuclear transcription factors and transcription factors as a whole. So now I will move toward the next problem. So problem number 5, it says that the HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for the HIV protease over mammalian protease. Number C, only symmetrical substrates are clipped by the HIV protease. Number D, there is no significance as such. So read the problem statement carefully, which says that the HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? I want everybody to try to answer this question. It is very easy. If you read the question and the answers carefully, you will be able to answer it. And I also want everybody to write in the chat box which of the following points or which of the following answers you think is the correct. What significance this thing might have? The HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by HIV protease. Number D, there is no significance. So, is there any answers for this question? Number B. So, everybody thinks it is number B. Okay, let us see. So, correct, it may be possible to design inhibitors that prove selective for the HIV protease over human protease. So, as I have mentioned earlier that the HIV protease enzyme is very important for the replication for the same. Okay, and HIV protease inhibitors are a very important class of drugs which are employed for the treatment of the HIV AIDS. Okay. So, HIV protease enzyme is symmetrical dimer made up of two identical protein units, each consisting of 99 amino acid. The active site for at the interface between the protein units and is also symmetrical with two-fold rotational symmetry, means C2 symmetry. The enzyme has a broad substrate specificity and can cleave a variety of peptide bonds in viral polypeptide, but crucially it can cleave bonds between the proline residue and an aromatic residue phenylalanine or tyrosine. Moreover, the symmetrical nature for the viral enzyme and its active site is not present in mammalian proteases, again suggesting the possibility of the drug selectivity. So, drug selectivity is a very important thing when it is comes to the drug design, because when you are targeting a thing which is a virus or a protein, as you can see, or a enzyme like kinase or a bacteria. So, the mechanism is very much related to our own body mechanism. Okay. So, these things the selectivity is very important, means if you are designing a protease inhibitor for HIV virus, the inhibitor must not bind to the mammalian proteases. Okay. Or if you are designing a kinase inhibitor for a cancer cells, the kinase inhibitor must not bind to the kinases of the normal human cells. Or if you are designing a drug to kill some part of the bad bacteria which are present in our body, it must not act on the good bacteria which are in our body. Okay. So, drug selectivity is a very important thing when you are designing a drug, particularly in medicinal chemistry, and there are many things actually for which you have to take care while designing the drugs, or and in our actually in the nature it is also like these things are designed in such a way, but you have to find out what is the particular difference where you can just work on. Okay. So, this was the problem number 5. Now, I will move toward the problem number 6. Again, I have said you, if you have any doubt regarding anything, any question, you can write in the chat box. Okay. So, problem number 6, it says that imatinib is an important anti-cancer drug which targets a protein kinase. What role does the piperazine ring has? This is the piperazine ring which is in the blue. Okay, and this is the molecule, the drug imatinib, which inhibits a protein kinase. And you have to find out and you have to Now tell me correctly, what role does this piperazine ring have? Number A, it increases the water solubility. Number B, it increases target selectivity. Number C, it increases target binding. Number D, all of the above. I will give you a brief about the protein kinases, how the kinases work and many things, because these are very important topic and very new topic to you, but this is very interesting because these are very much new drug therapies which have been employed in starting from the 2000s. Okay, because and this imatinib drug, it is the first, I think, approved drug for the kinase inhibitor, and it is for the anti-cancer drug. So, write in the chat box which of the following options you think is correct regarding the role of this piperazine ring in imatinib. Okay, so are there any answers for this question? Number D, number D, number D. So, I think everybody think it is number D. Okay, let us check what is the correct answer. Yeah, so very good. The correct answer is number D, all of the above. So, imatinib, now let us before going directly to imatinib and kinase inhibitors, now let us know what kinase does. So, basically protein kinases is a important enzyme of our body. So, what kinases do? The kinase enzymes, they take one phosphate group from the ATP and then it adds up to the proteins. Okay. So, if you have a protein, particular protein, let us say P, and or a particular protein, and I will just try to write it. So, if you have some protein, okay, and I will say write as pro, and if there is a if you have ATP, now what will happen, this kinase enzyme, what they will do, they will attach or phosphorylate the protein with a phosphate group taken from the ATP, and ATP will be converted to the ADP. Okay. So, the kinases, the work of kinase is to phosphorylate the particular proteins. And how they do it? So, as we know, there are some amino acids which have some hydroxyl group like serine. Okay, we have some amino acid which have the hydroxyl group like serine, then we have, I think, tyrosine, and we also have this kind of serine tyrosine. Okay, and as we can know that how they will phosphorylate, they will just make it like O and then Pi. Pi means the monophosphate from the ATP. Okay. And there is threonine also, serine, threonine and tyrosine. So, based on these three amino acids, how the kinase are divided, like it is the serine threonine kinase, or this is the tyrosine kinase. Okay. So, the amino acid residue for which it the particular kinase is phosphorylating, based on that the kinases are named. Okay. So, there is a particular enzyme substrate which is a BCR-ABL, and this is a which is expressed in the cancers also. Okay. So, what will happen, as you can see, the ATP is converted to the ADP. So, there is a ATP here. BCR-ABL is a protein substrate. Okay. So, is a particular protein which is targeted by this imatinib. Okay. So, that is why it is called the target is the BCR-ABL particularly. So, ATP is converted to ADP as you can see here, and the substrate it is get phosphorylated, and after the phosphorylation of the substrate I think target turns on a signaling, and the signaling is very important for the survival of the cancer cells. Okay. Now, what will happen if there is a imatinib? So, what is the role for the kinase inhibitor, how the kinase inhibitors work by? So, kinases are particularly a protein. So, as you can see there is a protein like in the blue chain, okay, and how these things comes and these structures were formed, this is the part of molecular docking, okay. And if you want I can just discuss it in detail in the last class, our class number 12. If you want I will just can show you how these things work, that like this docking and how these protein structures and are formed, okay. So, as you can see there is a protein structure, and there is a particular part of a protein now which is called as a substrate binding area, and the substrate is the ATP. So, there is a particular part of the protein where the ATP get binds, and from that ATP the phosphate is cleaved off, okay, and this part which is called the hinge region of the kinases. So, for a compound to be a kinase inhibitor, it must bind to the hinge binding moiety for the particular kinase, okay, or for the particular protein, okay. So, imatinib it works like that, it will just ATP binding pocket in the particular kinase, there is a ATP binding pocket, and the kinase inhibitors work is to go and bind in that ATP binding pocket. So, no ATP can go there and bind, okay, and that is how what will happen, there will be no phosphorylation of the substrate and there will be no signaling, and that is how the cancer cells will not able to survive, and this is the mechanism how the kinase inhibitors work. Now, not only the cancer, there are many I mean kinase inhibitors are mainly used in cancer and different type of cancers, but this kinase inhibitors also actually used in many kinds of other diseases, okay. So, as you can see this is the first kinase inhibitor which is approved by FDA in 2000 or 2001, I think, I do not remember exactly, but in early 2000s, okay. So, this was the first kinase inhibitor which is approved, and till now I think there are around 80 kinase inhibitors which are approved by various drug governing bodies, okay. So, this is the structure of imatinib, and this there are many roles of this each and every group, okay, and as you can see there is a this pyridine ring, there is this pyrimidine ring, okay, and all of these hydrogens and oxygens are bind to a particular group or particular amino acid of the proteins, and the piperazine ring, the main role of this ring is to increase the water solubility, to target selectivity and target binding. Okay, and as you can see this is the protein, this is the structure, this is the ribbon structure of the protein, and in the pink there is the kinase inhibitor imatinib, and the green are the amino acid residues. So, this part of the imatinib it goes to the adenine pocket, and adenine pocket means the ATP binding pocket, or we all know that adenine is a part of the ATP, and that where if there is a adenine pocket or ATP binding pocket, this group fits there. This group, this middle group, it goes to the hydrogen hydrophobic pocket, and this goes to the allosteric pockets, and there are many pockets. This is also in the detail, you do not need to know the so much detail about the kinases till now, but I think this was about the protein kinases which I gave you the brief. And if you want to know these things in detail, I can just discuss in the last class. So, in our last class, which will be our week 12 live session, so we will complete our, so today we are discussing week 11 course contents, right. So, in the next class, which will be your week 11 live session, there we will discuss the week 12 course content. So, for the last session, I want to just for the week 12 live session, so I will want to just summarize everything which we have read till now, and if you want to know some additional things like how the molecular docking has been done, or how these things, so you can just write me in the chat box, so that is accordingly I will just prepare my slides, okay. And if you have any problem for a particular topic in previous classes also, you can tell me in the next class or in today's class, so that I can just prepare the PPTs accordingly, okay. So, now I will move toward the next problem. So, problem number 7, it says that which of the following statements is false regarding the characteristics of a good protein target for antiviral drugs. Number A, it should be important to the life cycle of the drug. Number B, it bear little resemblance to the human protein. it should be important to the late stages for the virus life cycle, and number D, it should be common to different type of viruses. So, it will be life cycle of not drug, it will be life cycle of the virus, I think. Okay. So, there is a typographical error. Okay, I will just correct it later. So, which of the following statements is false regarding the characteristic of the good protein target for an antiviral drug? Number A, it should be important to life cycle of the virus. Number B, it should bear a little resemblance to the human protein. Number C, it should be important in the late stages of virus life cycle. Number D, it should be common to different types of the viruses. Write in the chat box which of the following you think is the correct answer. Okay, so is there any answers for it? Number B and number A. So, we have two answers, right? Okay, let us check what is the correct answer. So, the correct answer is number C. It should be important in the late stages of the virus life cycle. So, antiviral agents or antiviral drugs, so they can work by two manner. Okay, they targeting viral function or targeting cellular function that the virus needs. So, the first one, the targeting viral function may be the nucleic acid polymerase, protease, integrase or neuraminidase. So, these are the targets which can be targeted for the viral functions. Okay. And the first one, nucleic acid polymerase, it can be DNA dependent DNA polymerase, RNA dependent RNA polymerase or DNA dependent RNA polymerase. So, key characteristic for the antiviral drugs, able to enter the cells infected with the viruses, interfere with the viral nucleic acid synthesis and or regulation. Some drugs interfere with the ability of the virus to bind the cell. Some drugs stimulate body's immune system. Best responses to antiviral drug are in patients with competent immunity system. A healthy immunity system works synergistically with the drug to eliminate or suppress viral activity. So, the false statement regarding the characteristic of the good protein target is number C. It should be important in the late stages of the viral life cycle. Okay. And I think it is it will be for the life cycle of the not drug, it will be of the virus. Okay. So, always be careful when you are answering some true false kind of question, as I always have said, like these questions are somewhat tricky. If you are not concentrating enough, if you are not reading the question carefully enough, you will make a mistake surely. Okay. So, when there is a true false kind of question, please be careful. So, now we will move toward the problem number 8, which says that the following structure belongs to a group of compounds obtained from the African bush willow and shows a promising anti-cancer agent. Okay. Which of the following statements is not true? Number A, the above structure being tested as a phosphate pro-drug cancer chemotherapy. Number B, it prevents angiogenesis. Number C, the cis-trans isomers equally effective. Number D, it bears the structural similarity to colchicine. So, this drug you have to find out which of the following statement is not true. means which of the following statements is false regarding this particular natural product which is a promising anti-cancer agent. Number A, the above structure being tested as a phosphate prodrug in cancer chemotherapy. Number B, it prevents angiogenesis. Number C, the cis trans isomers are equally effective. Number D, it bears structural similarity to colchicine. Which of the following you think is not correct regarding this particular drug? So, are there any answers for it? Okay, so number B, number A, number C. So, we have 3 answers. Okay, B, A and C. Okay, let us see what is the correct answer. So, the correct answer is number C. The cis trans isomers are equally effective. This is the this statement is not true. Okay. So, this particular compound, it is called the combristatin, and combristatin is a very important natural product which works as a anti-cancer drug. So, it actually blocks the polymerization of the tubulin, and from the tubulin, in the cancer cells, from the tubulin the microtubule polymerization happens, okay, and this thing is blocked by this combristatin. As you can see, this is VEGF or VGRR-HIF, and it inhibits the angiogenesis, okay, and also when there is this microtubule polymerization is blocked in the cancer cells. What are the effects which it has in the cancer cell? So, cytostatic effects is the anti-proliferative effects, inhibit the spindle production, inhibits the cell cycle, inhibits the cell migration, apostasis or the viable cell shrinkage, chromination, condensation, membrane blabbing, nuclear collapse, apostatic body formation and apostatics. Okay, sorry. So, these are the things which are which happen if the microtubule polymerization is inhibited. And as you have seen, like the cis trans isomer, so this is the cis isomer, right? Now, this is the double bond and the 2 groups are at the same position, so this is the cis isomer, and there is also a trans isomer, but the trans isomer is not as active as the cis isomer. Okay. So, there is a difference. So, this statement which has said that the cis trans isomer have same activity is the false. Okay, and as you can see, combristatin has a very simple structure, but it is a very important, and many anti-cancer drugs actually have this particular functionalities of particular moieties, and which are formed during the pharmacophore hybridization methods. Okay. So, this was about the anti-cancer drug combristatin, which is also a natural product. So, problem number 9, it says that anti-ulcer drug omeprazole is number A, reversible proton pump inhibitor, number B, antagonist of H plus K plus ATPase, number C, agonist of H plus K plus ATPase, number D, irreversible proton pump inhibitor. Omeprazole, I think everybody have heard the names of this kind of drugs, because it is very common drug which we all use at some point of the indigestion related problem. If you have problem related indigestion or something, we always use this these drugs. Okay. So, you have to find out which is the correct statement or the correct classification of this particular anti-ulcer drug omeprazole. Number A, reversible proton pump inhibitor, number B, antagonist of H plus K plus ATPase, number C, agonist of H plus K plus ATPase, number D, irreversible proton pump inhibitor. so we have answers number C, number C. Okay, let us check what is the correct answer. So the correct answer is number D, irreversible proton pump inhibitor. Okay, so omeprazole is a irreversible proton pump inhibitor. Now this is the structure of the omeprazole, and as I have shown you earlier, I want to know what is the name of this particular heterocycle. This particular heterocycle, can you write in the chat box what is the name of this particular heterocycle which is present in this drug omeprazole? I have mentioned it earlier that there is you need to remember some a few heterocyclic when you are reading the medicinal chemistry, because the question can come directly from there. So what is the name of this particular heterocycle? Does anybody know? If you know, you can write in the chat box. Benzimidazole, very good. So as I have shown you what is the structure of imidazole, like this is the structure of imidazole, right? There is a nitrogen 1 and 3 position and a 5-member ring, but if there is a benzene ring which is fused with this imidazole, it is called the benzimidazole. Okay, and it is also a very important azole class of compound. Okay, so what is a proton pump inhibitor? So proton pump inhibitor or PPI are a group of drugs whose main action is a pronounced and long lasting reduction of the gastric acid production. Okay, proton pump inhibitors mechanism: omeprazole and lansoprazole inhibit the proton pump effectively irreversibly, requiring the synthesis of the new enzyme protein. Omeprazole and lansoprazole approved for the treatment of the duodenal ulcer, which may be conjunction with the triple therapy, erosive esophagitis, gastric acid hypersecretory state including Zollinger and Ellison syndrome. Okay, gastrinomas that cause secretion of the large amount of the acid. So this drug, anti-ulcer drug omeprazole and which is known as the Prilosec, and lansoprazole which is known as the Prevacid, these are the irreversible proton pump inhibitor. Okay. Now we will move toward the problem number 10. So problem number 10, it says that the following structure saquanavir is a protease inhibitor. Okay, and the decahydro isoquinoline system in the protease inhibitor was used to replace an amino acid residue that is present in the lead compound. Which amino acid was replaced? Number A, phenylalanine, number B, aspartic acid, number C, proline, number D, tryptophan. The following structure saquanavir is a protease inhibitor. The decahydroisoquinoline ring system in the protease inhibitor. This is the decahydroisoquinoline ring in the protease inhibitor was used to replace an amino acid residue that is present in the lead compound. Which amino acid was replaced? Number A, phenylalanine, number B, aspartic acid, number C, proline, number D, tryptophan. Protease inhibitors, just I have mentioned earlier, this saquanavir is a very important HIV protease inhibitor. Okay. So is there any answers for it? Number B. Okay, let us check what is the correct answer. So the correct answer is number C, proline. Okay, once again. So saquanavir is sold under the brand name Indiviores and photovars. Okay, so socquinavir binds to the active site of viral protease and prevents the cleavage of the viral polyproteins. So, as you can see this structure, if this decahydroisoquinoline, and as you know what is the structure of the, if you know remember the structure of the proline, you can understand how the structures are related to each other. Okay. So, in the proline we have this kind of ring. Okay. So, as I know, in the proline we have this kind of ring amino acid, and proline is the only amino acid which is I mean has a secondary hydrogen, right. Okay. So, as in there is a H also. Okay. So, as you can see the structure is very common, like if there is a N nitrogen here and at the alpha position there is a carboxylic acid group and it is cyclic. So, that is how you can just understand how there is a structural similarity, and the lead compound is first designed and then it is modified to have the particular compound. Okay. So, there is some things. Okay. So, as we can start here. So, if we divide this particular drug in many places. Okay. As you can see there are multicolor have been used. Okay, here. So, let us start from this place. So, P1 ligand. So, P1 ligand means this part. Okay. This in the this decahydroisoquinoline ring, which is this P1 pro has been replaced. So, P1 pro, so there was a proline at first here. Okay. In the lead compound. So, this has been replaced by a bicyclic decahydroisoquinoline in socquinavir. In other protease inhibitor, different hydrophobic groups have been used like phenyl tertiary leucine, tertiary butyl. Use of the sulfonamide and carbamate link to P1 ligand improves the pharmacokinetic properties, including better oral availability. Now, this is the P2 prime ligand. So, different hydrophobic alkyl aromatic groups have been tolerated here. Cis amino hydroxylin most moiety in the EDV or it is indonavir led to the 100-fold increase in potency compared to the benzylamine analog. Thiazolol moiety RTV increases solubility, benzamide derivative bearing substituent in para position like NH2 OMe OH are able to form hydrogen bonds are optimal for potency. So, this is the how the molecule, this molecule is optimized, how this molecule is the SAR have been performed. So, these are mentioned in this slide. Okay. So, like this peptide bond, okay, this P1 prime peptide bond translation state binder, which is hydroxyl group, is crucial to mimic the catalytic transition state, forming key hydrogen bonds with carboxylic acid group of aspartate 25 and aspartate 25 prime. Hydroxyethylamine, hydroxyethylene, hydroxypentene as a hydroxyethylamine are usually used as the translation state mimic moieties. P1 ligand, which is in this yellow or yellowish orange. Okay. So, unsubstituted benzyl group is preferred here. Larger groups like pyridyl methyl piperidine moiety in indonavir interact with the S3 side. P3 ligand, which is in the green, large hydrophobic group can accommodate in this subunit in the following generation of protease inhibitors. P3 ligand has been removed because it is not necessary for the activity interaction outside the substrate envelope. And this purple thing, this is the ASN or asparagine residue was replaced both in reduced peptide character and increased binding affinity. Carbamate and uridolinker increases the metabolic stability. Good ASN mimics the thiazolide methyl moiety in RTV, cyclic urea in LPV and tertiary butane in IDV. This RTV, LPV and IDV, these are the various this protease inhibitor names of the like ritonavir, lapatinivir and this indonavir. Okay. This THF substituent DRV increases the binding affinity, strong hydrogen bonding, van der Waals interaction to the S2 subunit. So, that is how the SAR is happened in this moiety. Okay. By changing each and every group, we have to see like how the activity happens, and that is how the molecular lead compound is diagonized, okay, for the particular disease, and you do not need to remember all of this thing. I just have shown you to for your better understanding. So, now we can move toward our next problem. Problem number 11, it says that the following structure is a cephalosporin prodrug of an alkylating agent. Which enzyme, what enzyme can be linked to the antibody to release alkylating agent? Number A, carboxypeptidase, number B, beta lactamase, number C, aminopeptidase, number D, esterase. So as you are on this problem number 11, okay, so I think we can like have a 2-minute break. Okay, so I will give you break of 2 minutes. Till then you just look at the problem number 11 more carefully and write in the chat box which of the following you think is the correct answer. I will just keep my camera off, but I am in here, okay, and after just 2 minutes I will rejoin. Okay, so I am here. I will just start the session again after 2 minutes, and you don't leave, but read the problem number 11 carefully and write in the chat box which of the following you think is the correct answer. Okay. Okay, hello. Can you hear my voice? So is it audible to you? We will restart our session then. Okay, very good. So there we have, I think, 1 or 2 answers. So let us check out what is the correct answer. So the correct answer is number B, beta lactamase. Okay. So cephalosporins are a group of semi-synthetic antibiotics derived from the Cephalosporin C, obtained from a fungus Cephalosporium acremonium. So mechanism of action: cephalosporins are a bactericidal and have some mode of action as the beta-lactam antibiotics such as the penicillin. Cephalosporin disrupt the synthesis of peptidoglycan layer of the bacterial cell wall. The peptidoglycan layer is important for cell wall structural integrity. Okay. So as you can see, there is a beta lactam ring present here, right. So this enzyme which is which can be linked to the antibody to release the alkylating agent. Okay. So this that can be is the beta lactamase. So when there is a structure, so you look at carefully the structure to know what is the common area. So as we all know the structure of the penicillin, and cephalosporin has a similar structure, a little bit different, but more or less similar structure because it has a beta lactam ring which is present here. Okay. So, now problem number 12, it says that method of action of a aromatase inhibitor is number A, irreversible inhibitor, number B, reversible inhibition, number C, suicide inhibition, and number D, uncompetitive inhibition. So, aromatase is another, okay, I think they are. So, aromatase is another important target for many drugs. Okay. So, the method of action for aromatase inhibitor is, I think there is something error. Okay. So, if you have any doubt, you can just write in the chat box. Okay. If you have any problem, any doubt, please write in the chat box. So, method of action of aromatase inhibitor is number A, irreversible inhibition, number B, reversible inhibition, number C, suicide inhibition, number D, uncompetitive inhibition. So, is there any answer for it? Number A. Okay, so we have only one answer. Okay, let us check out. So, the correct answer is number C, suicide inhibition. Now, what is aromatase inhibitors? How the aromatase inhibitors work? Okay. So, this is an important class of the anti-cancer drug again. Okay. So, as you can see, there are testosterone and androstenedione, okay, as you will say, androstenedione, okay, sorry. So, it is a kind of hormone. So, from this hormones estrone and estradiol are formed, and the aromatase is the enzyme which actually catalyzes the formation of the estrone and estradiol. Okay. And what will this hormones will have? The estrone and estradiol, they will activate the ER or generation of the E2-3,4 quinone. Okay, the first one, the proliferation of the mutated cell, and the second one is required for the DNA alkylation or the depurination, and the cancer is the result for this mechanism. So, aromatase inhibitors, they actually inhibit this aromatase particular enzyme. So, these are the very important class of the drug for which acts as anti-cancer drugs. So, as you can see, this is the structure of mechanism for the aromatase inhibitor. There is a brief structure and how this and a particular inhibitor works. Okay. And as I have always said that you do not need to remember the mechanism in detail for now. Okay. But this is all only for your knowledge. I have just given this mechanism in brief detail. Okay. So, as you can see in the aromatase, there is this is the active site. Okay, there is a Fe2 active site, which is a prism kind of structure, and when there is a presence of oxygen, it forms the Fe3 and OO radical. Okay, and there is single electrons transfer, for which this O minus has been formed, and it attacks to this particular substrate, and that is how the catalytic cycle goes on. Okay. And this aromatase inhibitors, as I have said, so this mechanism is the suicide inhibition mechanism. Okay. So, now problem number 13, it says that what sort of receptor is the muscarinic receptor? Muscarinic receptor. Okay. Number A, G protein couple receptor, number B, kinase linked receptor, number C, intracellular receptor, number D, ion channel. I think these receptors and this muscarinic receptors we have discussed earlier in our some previous, I think in first classes, which we have discussed in, I think, class number 2 or 3, we have been discussing these receptors. Okay. Now, you have to identify what kind of receptor is this muscarinic receptor. Number A, G protein coupled receptor, number B, kinase linked receptor, number C, intracellular receptor, number D, ion channel. Is there any answer for it? Option A. Option A. So we have two option A's, right? Okay, very good. The correct answer is number A, G protein coupled receptor. Now, how G protein coupled receptors work? G protein coupled receptors or GPCR in the abbreviated form are the largest class of membrane proteins in the eukaryotes. They mediate most of the physiological responses of hormone, neurotransmitter and environmental stimulants. So, when a GPCR is not bound to an agonist, it remains inactive. The G protein also remains inactive on the cell membrane. Upon binding of a ligand or agonist such as hormones or neurotransmitters, the GPCR undergo a conformational change, activating its GEF domain. GEF domain activates the monomeric GTPase to replace GDP from a GTP in a GS alpha domain, activating G protein. Upon activation, the GS alpha domain dissociates from the GPCR G protein complex and binds to the effector enzymes on the cell membrane to activate it. The activated effector enzyme generates second messengers that activate intracellular signal transduction cascades. The hydrolysis of the GTP to GDP in the GS alpha domain dissociates the GS alpha domain from the effector enzymes, deactivating the enzyme. So, this is how the G coupled protein receptors work. And muscarinic acetyl coding receptors. So, these receptors are classified from M1 to M5. Okay. So, the M1 it is the works on CNS or salivary glands or parietal cells, okay, and CNS excitation, memory, locomotor activity, gastric acid secretion, this is increased by this receptor. M2 it works on the heart and decreases the CAMP, so the heart rate, force and AV conduction, and M3 it smooth muscle or exocrine gland. So, all smooth muscle contraction except vasodilation and gradual secretion is increases. Okay. So, this is about the G protein coupled receptors. So, let us go to next problem. So, problem number 14 says that which of the following statements is true regarding HIV protease enzyme. Number A, it belongs to enzyme family of the cystinyl protease. Number B, it catalyzes the formation of peptide bond. Number C, active site structure contains cysteine moiety. Number D, active site structure contains aspartic acid. So, as I have said that HIV proteases are a very important enzyme and a very important target for developing anti-AIDS drugs. Okay. So, you have to find out among these 4 options which is the true regarding HIV protease enzyme. Number A, it belongs to cysteine family of cystinyl protease. Number B, it catalyzes the formation of peptide bonds. Number C, active site structure contains cysteine moiety. Number D, active site structure contains aspartic acid. So, I want everybody again to try to answer this question. It does not matter if it is wrong or right, but at least you should try. So, is there any answer for this question? Number B. Okay, only one answer. Okay, let us see. Okay, so the correct answer is number D. Active site structures contains aspartic acid. Okay. So, what is the function of the HIV protease? So, one long chain of HIV protease proteins inside the infected cell. This is as you can see in the red. This is the one long chain of HIV protein inside the infected cell. Okay. And what does the protease does? Protease actually makes this long chain into two shorter chain of the HIV protein, and individual proteins which are formed, which also can make new HIV particles in individual enzymes that help will beat new HIV particles. So, that is how the HIV protein it basically replicates. And what does this protease inhibitor do? This molecule will stop this protease so that there are no new HIV proteins. Okay. So, this is the active site structure. As you can see, the active site structure of this protease enzyme. So, all we know that protease is an enzyme and all enzyme have an active site. Okay. So, as you can see, there is an active site structure and there is a aspartic acid residue. Now, how this aspartic acid residue works? As you can see, there is a enzymatic cycle here, and this aspartic acid stays as a aspartate, and there is a hydrogen bonding between this carbonyl carbon and this aspartate O minus here, and it will abstract the hydrogen from a water molecule here, and this will make this hydroxyl which is a good nucleophile. Now, this will attack on this carbonyl carbon of this structure. Okay. And what will happen after attacking this carbonyl carbon? It will form a tetrahedron carbon here. Okay. Now, what will happen again? This again another aspartate residue, it will abstract the proton from this hydroxyl group, and it will just cleave this molecule, this protease. So, that is how this is the protein moiety, and that is how this enzyme which in the inactive site which have this aspartate, it will cleave the moiety. Okay, as you can see, there is a cleavage. This was from one protein chain here, one peptide bond here, and it is cleaved by this particular aspartate residue of the active site of this enzyme. Now, what will happen again? This catalytic cycle, how it will finish? So, as you can see, this aspartate it will again this can actually work by two phases. Okay, it can also reversibly cleave the protein, or it can also have this kind of reversible cycle, and which will give us the another action protein. Okay. So, what can happen? As you can see, there is a this kind of mechanism are studied by the isotope labeling experiments. Okay. And this, as you can see, this is the water. When I will share this PPT, you can just look at this PPT more carefully. There is a water molecule present here which has the O18. Okay, O18, and this O18 isotope, and it will when this it can actually cleave this molecule, or it can reversibly incorporate the O18 of this oxygen of this water to this peptide linkage, and it can just have this reversible kind of moiety. Okay. So, I think you do not need to remember or memorize exactly how the mechanism works, but remember this aspartic acid is present in the active site. Okay, in the mechanism, I do not think you need to remember the all detailed mechanism, and these are few examples of the protease inhibitors. Okay. So, as I have mentioned earlier, like this protease inhibitor, this is the indinavir, this is nelfinavir, this is ritonavir, this is saquanavir, this is amprinavir. Okay. So, how to remember what is a protease inhibitor? This I think this ending of this is the VIR. Okay, AVIR. So, whenever there is this present this NAVIR, whenever there is a term present as the end of the drug, so you remember it is a protease inhibitor. Okay, and as you can see the structure, it is a very much there is a many peptide linkage and protein linkage kind of amide linkage. So, this is a one more important aspect of the protease inhibitors. Okay. So, now next problem number 15, it says that palbociclib is an inhibitor of dash kinase. Number A JK, number B BCR-ABL, number C CDK 4 or 6, number D MET. So, all of these are a particular class of the kinases, as I have mentioned earlier, like kinase inhibitors. What is the kinase enzyme? I have mentioned in the previous one question. So, there are many family for this kinase enzymes, okay, like this JAK, BCR, ABL, CDK 4 or 6 or MET kinase. So, these are various families of the kinase enzymes, and each and every family has some important work, okay. So, this palbociclib is a particular inhibitor of a family, and you have to identify which of the kinase family it belongs to, okay. And as I have mentioned imatinib in previous, so that was an inhibitor of BCR ABL, okay, class of kinase. Now, palbociclib is another inhibitor which is very important anti-cancer drug. So, try to answer in the chat box which of the following you think is the correct. So, is there any answer for this question? Number C. Okay, so we have one answer which is say it is number C. Okay, let us check. So, it is, yeah, correct, it is number C, CDK 4 or 6. So, CDK stands for the cyclin dependent kinase. Now, so how this compound, as I have said you, so let us discuss a little bit. So, palbociclib, it is known as the Ibrans, so it is the commercial name. So, this is the structure for this particular kinase inhibitor. So, in the blue it is the pharmacophore, okay, which binds to the adenine pocket of the or ATP binding pocket of the particular kinase. So, this primary target is CDK 4 or 6, CDK stand for cyclin dependent kinase, okay. So, this compound is a CDK 4 or 6 kinase inhibitor. So, this is a type 1 inhibitor, and it binds to the ATP pocket in an active conformation through the 2 hydrogen bonds with the kinase hinge valine 101 and one with aspartate 1663 of the DFG motif, and a cyclopentyl group occupies the ribose pocket, this cyclopentyl group, okay. And DFG motif as okay, this thing I have not mentioned yet. So, in every kinase enzyme, okay, so there is a particular motif which is called the DFG motif, okay. So, and the most of the, I mean 99% of the kinases have this particular conserved motif, and this DFG motif stands for D for the I think D for the glutamic acid, and F for the phenylalanine, and G for the glycine. So, as you can when you will see there is a particular chain of this 3 amino acids all together, and this residue is conserved in most more or less all kinases, okay. And this is the motif, and as I have mentioned, hinge region is the very important region, which is for a molecule to be a particular kinase inhibitor, it must bind to the hinge region, okay. So, okay. So, treatment for a hormone receptor HR positive, HER negative advanced or metastatic breast cancer in postmenopausal women, and side effect of this drug is neutropenia and leukopenia, okay. So, how does now as I have mentioned you, like this kinase inhibitors act as a anti-cancer drugs. So, why it acts as an anti-cancer drug? because this cyclin dependence, so how this happens? So, CDK4/6 inhibitor, how they inhibit the cell cycle of a malignant cell. Okay. So, as you can see, this is the cell cycle, okay, and the first one is the G1. Okay, so it here cell accumulates the resources to needed to duplicate the genome. After G1, the cell can go to the S phase, or it can stop at the G0 state, where it is the quiescent state where the cell rests. Okay. And in the S phase, the actual duplication of the genome occur, means the DNA synthesis. Now, then it goes to the as you can see, this goes to the G2 phase, and in the G2 phase proper DNA replication checked and the final preparation of mitosis phase, and in the M phase, which is the last phase, is mitosis phase, where chromosomes are separated and actual cell division occurs. So, this is the cell cycle. Now, in the cell cycle, this CDK4/6 inhibitors, they actually block the G1 phase, okay, and will means CDK4 and CDK6, which is important for this G1 phase of the cell. Now, if the G1 phase is inhibited, so what will happen? The cell cycle will be interrupted and cell division will be decreased for the cancer cells. Now, you can say that there may be this particular enzyme can also work for the human healthy human cells also. And so, there is I have always said, like there is a form where the selectivity comes. Okay. So, in many kinase inhibitors, the particular cells which are the cancer cells and normal human cells, they have some differences in their cells and the nucleus and all those things. So, these changes, these differences are targeted by this kind of anti-cancer drugs. Okay. So, this was all about your problem number 15. Now, let us go to the next problem. Problem number 16, it says that omeprazole is a proton pump inhibitor, which I have mentioned earlier also. Which region is prone to the metabolism? Number A, number B, number C, or number D. So, as you can see, this is the structure of the omeprazole. Okay. So, this blue one is the number A, this methyl group, this OMe group is number B, this C, this methyl is the number C, and this benzene is number D. So, which of the following region is prone to the metabolism? Write in the chat box which of the following you think is the correct answer. So, do we have any answer for it? Number B. So, we have number A and number B. So, we have two answers, number A or number B. Okay, let us check. So, the correct answer is number A. Okay. So, omeprazole, as I have mentioned earlier, it is a, okay, let us move toward the next phase. Okay. So, it is a irreversible proton pump inhibitor, right. So, as you can see here omeprazole, okay, it actually reversibly enters to the cell. So, this is the blood, this is the cytosol, and this is the secretory region. Okay. So, in the so here the pH is 7.4, here the pH is 7.1, here the pH is less than 2.0. Okay. So, this is the omeprazole here. Omeprazole, it will go to the cytosol, and in the when it is region this secretory region, and the omeprazole, it will there is sulfonamide, and this SS sulfonamide, which will inhibit this H plus ATP K plus ATPase. Okay. So that's how the particular this omeprazole actually works. Okay. So in the blood there is omeprazole, okay, and their pH is 7.4 and partial parietal cell canaliculus lumen. Here the this pH is 2.0. So what will happen when the pH is 2.0 means this acidic pH, means we have H plus ion. Now this H plus ion, it will attach to this nitrogen here, okay, this nitrogen here, this on the pyridine ring, and after 3 steps this sulfonamide intermediate will be formed, okay, and then we will have this H plus A plus KTPase, and which have its thiol group, and it will attach with this SS bond, and in enzyme inhibitor complex is formed, okay. So as you can see, there is a gastric gland lumen is present here, wherein there is a acid of HCl, and that's how this is the cycle, how this thing happens, okay. And there is a acetylcholine receptor, muscarinic receptor antagonist present here, there is a gastrin present here, and this calcium dependent process. So that's how the H plus K plus ATPase works by, okay. And proton pump inhibitor, they actually inhibit this particular enzyme, and by this it acts on the ulcers, peptic ulcers, okay. So number A is the correct answer. So now next problem. So problem number 17, it says that the DNA synthesis checkpoint is present in dash phase of the cell cycle. Number A M phase, number B S phase, number C G1 phase, number D G2 phase. So DNA synthesis checkpoint is present in dash phase of the cell cycle. I have just shown you in last to last slide what is the cell cycle, particular cell cycle, and how the cell cycle goes. So you have to identify there are 4 phases like G1 phase, S phase, G2 phase, M phase, and there is also a phase which is called Z0 where the cell rests. So there is, in the particular phase, there is a DNA synthesis checkpoint is present, okay, and this checkpoint actually checks if the DNA if the cell is capable of synthesizing DNA with which is ideal, okay, and the conditions are perfect. So it also checks this. If not, then the cell will rest, otherwise it will go. So you have to identify which is the correct phase where the DNA synthesis checkpoint is present. M phase, S phase, G1 phase or G2 phase. So do we have any answer for this? Number C. Okay, let us check. So the correct answer is number B, okay, S phase. So this is the following diagram which describes what different checkpoints of the cell are responsible for. So as I have mentioned earlier, there is G1 phase, and in this is the cell growth checkpoint, okay. So what happens in the G1 phase? So there is a cell growth checkpoint occurs toward the end of the growth phase of the or G1 phase, okay. So this is here the cell growth checkpoint is happen in the G1 phase. Checks whether the cell is big enough and has made the proper proteins for the synthesis phase. If not, the cell goes to the resting phase G0 until it is ready to divide, okay. Now, DNA synthesis checkpoint, it is present or occurs during the synthesis phase or S phase, okay, here, and it checks if the DNA has been replicated correctly. If so, then the cell will continue on to the mitosis. And the third checkpoint is the mitosis checkpoint. Mitosis checkpoint occurs during the mitosis phase M. Check whether the mitosis is complete. If so, the cell divides and the cell cycle repeats. Okay. So, there are 4 phases G1, S, G2 and M. There are 3 checkpoints: cell growth checkpoint, which is present at the end of the G1 or growth phase, DNA synthesis checkpoint, which is present in the S phase or DNA synthesis phase, and mitosis checkpoint, which is present in the M phase or mitosis phase. Okay. Cancer can result when one or more of these checkpoints malfunction. Okay. So, what happens for the cancer, the cell never goes to the G0 state. Okay. It will, if it is not even capable, it will all go on replicating itself, and the G0 the resting step does not happen. Okay. And if our drug can inhibit one or two phase of the cell cycle, it will act as a anti-cancer drug. Now, next problem. Problem number 14, it says that David Baltimore, Nobel Prize winning biologist, classified viruses into dash groups. Number A 4, number B 6, number C 7, number D 8. The viruses are classified by this Nobel Prize winning biologist David Baltimore in few groups. So, you have to identify how many of the groups he have classified the viruses into: 4, 6, 7 or 8. Write in the chat box which of the following you think is the correct answer. So, do we have any answer for it? Okay. Number B. Okay, so we have one answer, number B. Let us check. So, the correct answer is actually number C, 7. Okay, so these are the classifications. Okay, so basically the total there are 7 classes. Okay, and the classes are so basically the biologists have classified the viruses in 7 classes, but there are initially 3 broad classes. Okay, the first is DNA viruses, RNA viruses and reverse transcripting viruses. So, for the DNA viruses, these are group 1 and group 2. So, in group 2 there is a double stranded DNA, and the group 1 and group 2 has single stranded DNA as their mainly genome, and this single strand DNA can also form double strand DNA. Okay, and the RNA viruses are 3 type. Group 3 has double strand RNA and as the genetic material, and group number 4 has single strand RNA, and group number 5 has single strand RNA negative. Okay, so these are positive, negative. These are the same things about this orientation. and reverse transcripting enzyme, there are single strand RNA positive and double strand RNA positive or negative. Okay. So, these are the 7 groups in which the Nobel Prize winning biologists have classified the viruses into. Okay. Now, problem number 19, it says that which of the following is not an anti-HIV drug? Number A, nevirapine, number D, didenosine, number C, dihydroartemisinin, number D, zidovudine. Read the names carefully. I think I have discussed this thing in some previous classes also. So, which of the following is not an anti-HIV drugs? Number A, nevirapine, number B, didenosine, number C, dihydroartemisinin, number D, zidovudine. So, is there any answer for this question? Number C, okay. So, we have 2 number C. Okay, very good, it is correct, dihydroartemisinin. So, these are the structure of the drugs. Okay, this is the structure of the nevirapine, this is the didenosine, this is the zidovudine. So, these 3 are the anti-HIV drugs. Okay, but dihydroartemisinin, as you can see the structure here, it is an anti-malarial drug. So, I have discussed like artemisinin and artemisinin combinational therapy, which are very much used in the malarial treatment. Okay, so this is the drug which is used here. Okay. So, last question of today's session, it says the only DNA antisense molecule that has been approved as antiviral drug is number A, nevirapine, number B, formivircin, number C, darunavir, number D, zidovudine. The only DNA antisense molecule that has been approved as antiviral drug is nevirapine, formivircin, darunavir, zidovudine. So, do we have any answer for it? Okay, number C, number B and number C. Okay, let us check which is the correct answer. So, the correct answer is number B, formivircin. Okay. So, what is the antisense oligonucleotide therapy? So, as you can see, there is a DNA, right? We'll just sorry. So, there is a DNA, and from the transcription DNA it will form mRNA, and this mRNA via translation it will form from disease associated protein. Now, this transcription, if there is a antisense drug, okay, so what will this antisense drug will do? It will form a antisense strand. Okay, and what is the antisense strand? Basically, there are 2 type of strands in the DNA. One is the sense strand, which have this genomic coding, okay, and another is antisense strand, which does not have it. Okay, so it basically when there is a antisense drug, it is an oligonucleotide which will form this kind of DNA, and RNAs degrades RNA, no translation will happen. Okay, and this will degrade and no translation will happen. If no translation happens, no disease associated proteins are produced. Okay, and that is how this formycin works. Okay, it actually acts as a DNA antisense molecule. As you can see, there is a this antisense molecule, how it bridges, and that is how this form this kind of thing. So, this molecule, it is represented like this. Okay, this sphere or stick modeling. So, this was the last question of today's session. So, today we will discuss about several anti-cancer drugs, several anti-cancer therapy, and we discussed also about various viruses regarding HIV proteases and HIV viruses mainly. So, this was all about your last week 11 class materials. Okay. So, today's session video and this lecture material I will just post in the NPTEL website. You can just check it out from there. And if you have any doubt, any query, as I have always said, you can just mail me, and I have just like every day I will share my email ID once more. So, this is my email ID which I am sharing in the chat box right now, and if you have any doubt, any query regarding any class or any class material, you can just drop a mail. Okay. And this was our week 10 session, and like every Saturday we will next Saturday we will also meet, and next Saturday we will discuss your last week or week 12 course materials. Okay. And one more thing, in the last class I want to summarize the whole course of the medicinal chemistry which for the NPTEL which we have discussed till now, and in that class I want to know, like if you have any particular topic, if you want me to revise, or if you do not understand in particular topic. So, I will just discuss that topic in the last class. So, you can just drop a mail to me, or you can just write in the next class also which are the topics you need to or you want me to repeat. That is accordingly I will prepare the PPTs. Okay. And I will share this PPT and today's class material soon in the NPTEL website. So, thank you for joining the class. I will meet you in the next week. Okay. So, now I will close the session for everyone. So, thank you.